First up is Thomas Tedder with Collective Biotherapy.
As a representative of Selective's management team, experienced management team, all of us with at least 2 companies under our belts, it's a pleasure to tell you what this Eshelman Ventures-backed therapeutics company is doing. Cancer has touched all of our lives. Last month, I lost a young brother-in-law and close friend to lung cancer. After relapsing after a conventional standard of care therapy, he went on to the new checkpoint inhibitors in the clinical trial that you see advertised on TV all of the time. But as with many patients, the toxicities were too severe and he had to withdraw from the trial. Selective is focused on developing new therapies that have minimal toxicities to provide advantages over current therapies in inflammation, autoimmune disease, and particularly cancer. Selective will do this by taking advantage of a discovery in our laboratory 10 years ago of a small subset of B cells called B10 cells that function like the brakes on your car to inhibit the development of inflammation in autoimmune disease. Selective has developed 2 first-in-class therapies that can either upregulate B10 cell function or inhibit B10 cell function. In the first case, we get rid of B10 cells by depleting them using monoclonal antibodies. And in mice with tumors, for example, this leads to robust anti-tumor immune responses that are as therapeutically effective as the current checkpoint inhibitors that I just talked about. Moreover, these B10 cell depletion works in combination with checkpoint inhibitors, and thereby we predict, due to its unique mechanism of action, will have less overall toxicities. I look forward to discussing this with you because Selective is humanizing its lead monoclonal antibody depletion antibody in May, and we will be raising a Series A financing to take this monoclonal antibody through manufacturing into an IND with clinical trials projected in 2020. You can find me at the table in the Innovation Room, number 27, where we have an exciting story as well as free cookies. Thank you.
Next is Ed Burgard of Dignify Therapeutics.
Thank you. Ed Burgard, I'm president of Dignify Therapeutics. We are a drug development company focused on restoring bladder and bowel function to individuals that suffer from incontinence and the inability to control bladder and bowel function. There are over 50 million people worldwide that suffer from urinary and fecal incontinence. These are severely disabling conditions that are associated with diseases such as multiple sclerosis, spinal cord injury, diabetes, and of course advanced age. In fact, one of the primary reasons for placement of the elderly into institutional care is incontinence. It's an $8 billion drain on the healthcare system, and most of that money goes to managed care or specifically to skilled nursing care, but also to very primitive management products such as adult diapers and bladder catheters. It's an area that's virtually unrepresented by pharmaceuticals, and that's where we come in. We're developing a new drug to produce on-demand emptying of the bladder and bowel for those individuals that can't control emptying of those organs. The concept is very simple. Our lead program is designed to produce on-demand voiding. So a simple concept, you take the drug, within 5 to 10 minutes, the bladder and bowel completely empty, thus restoring the ability of those individuals to control when and where they empty their bladder and bowel. A little bit about the company. We're a team of entrepreneurs. We have over 50 years of collective big pharma experience. We founded 5 separate startups previously. And for our lead program alone at Dignify, in the last 3 years, we've raised over $12 million in venture capital, as well as NIH awards. We have a very fast and economical development plan, and we're currently seeking investments to fund our lead program to get us to an IND filing and through phase 1 clinical trial for on-demand voiding. We're at table 23 in the Innovation Room. We invite you to stop by. Thank you.
Next is Zach Forbes with Seldom.
Thank you, you, CED, for putting on such an amazing conference. It's an honor to be able to speak to you all today. Seldom is a single-cell analysis company. Single-cell analysis is the study of complex biological systems with heterogeneity. This means you need to find, identify, and study rare events. And sadly, it's rare events in biology that makes many diseases effective at killing us. And they can occur at scales of 1 in 10,000, or in cases like HIV, even way higher than that. And sadly, to now, there are no existing product offerings that allow you the ability to, at the appropriate scale, phenotype and genotype your samples and have a unified data set that is truly actionable. This is why we founded Seldom. Seldom's product is going to consist of a simple benchtop box that will integrate with your existing microscope. We'll be able to process our single-use microfluidic chips. You can array tens to hundreds of thousands of cells, perturb them, stimulate them, culture them on chip for up to weeks, phenotype them as you will, do your library prep on the chip. After you sequence them, you can input your data back into our software, unify these data sets, and be able to have phenotype and genotype together at scale. Truly actionable workflow. You're going to love it. We are a Duke University company. I'm alumni, but I come to you from product development at Philips and BD. Thank you, Duke University, to the exclusive license of all IP from the labs of Dr. Ben Yellen of Mechanical Engineering and Dr. Chris Wood of Cancer Biology and Pharmacology. We are a pre-revenue company, but we are doing our first installation in the next couple of months. We have just received an Illumina sequencing grant, and we'll be going to their new facility in Foster City in April to collaborate on our genomics workflow. We are here starting a Series A following the close of a $300K seed round and our SBIR phase 1 that's been funding us to date. Our Series A will be used to integrate the product and begin our launch to start getting our product validation and key publications on the market for a global expansion. Thank you for your time. Enjoy the conference.
Now we have Shawn Hinchin of Falcon Therapeutics.
Good morning, everyone. Our team takes your skin and morphs it into stem cells that seek out and kill cancer. It's always been our mission, our passion, to create therapies capable of fighting the deadliest forms of cancers for which there's currently simply no cure. And thanks to the amazing team, the network that we've assembled, we're advancing groundbreaking discoveries in the basic sciences into therapies that can truly help patients. My name's Shawn Hinchin. I'm the co-founder of Falcon Therapeutics. And you heard me right. We're creating an entirely new approach to cancer by harnessing the power of stem cells. All we need is a little bit of your skin and a little bit of time. If you think about that, it's remarkable. That means all of us are walking around right now with our own personalized therapies for cancer. Just a little bit of your skin. So let me tell you how we do it. We're taking advantage of the latest in the world of stem cells, Nobel Prize-winning technology that allows us to morph your skin into any cell type we want. Our team discovered that you can do this fast enough and in a unique way that lets the cells morph into things that chase cancer. They can chase it down. And because we can load these with drugs, when they hone in on the cancer, they release the drugs right at the tumor for specific killing. Now, the very first indication we went after was glioblastoma, or GBM. It's the most common primary brain tumor and one of the deadliest forms of cancer. But our technology, our testing quickly showed that this is a platform applicable to a wide range of tumors. So we've been adapting this to show that we can treat adult and pediatric, liquid and solid, primary and mets. This is allowing us to develop a fully robust pipeline, and we're honing in on that lead clinical candidate. But all the cancers have one thing in common. The standard of care simply doesn't work. However, our initial testing, we showed that we can flip these cells. They can crawl through the brain and hunt down brain cancers. They can home through the body and kill mets and get dramatic killing. This is not only allowing us to develop a tool that can empower clinicians, but it's creating hope for patients. This is Falcon. Thank you.
Please welcome Gary Pace with Cell Microsystems.
Excuse my voice today. It's an early spring in North Carolina, as I'm sure many people recognize. Cell Microsystems is an early growth stage company that develops, markets, and manufactures innovative products for single-cell research. Single-cell research has recently been leading to advances in our understanding of cancer, immunotherapy, and genomics, as well as being the foundation for the Human Cell Atlas project. The cell raft technology from Cell Microsystems allows researchers to isolate single cells with high viability and unperturbed phenotypes even at samples too small for other traditional separation technologies. The researcher can seed, sort, image, analyze, and recover single cells on one array on one platform. There's no other product on the market today that has this set of features at our price point. The cell raft technology has been validated for RNA sequencing, single nucleus sequencing, CRISPR-Cas9 transfection, and T-cell screening, among other product applications. We've grown our customer base to include research labs from academia as well as pharma companies. We've grown the company through non-dilutive financing and have recorded net positive income for the last 2 years. We have an experienced team of scientists and engineers that develop our products and manufacture them in our facilities at the First Flight Venture Center in Research Triangle Park. For 2018, we're focusing on the initial sales of our first automated platform. We're also looking for a full-time salesperson and to expand our facilities, and finally, to launch new products for the gene editing market. We're here today at the conference looking for new collaborations and strategic alliances for our products and our product applications. If you haven't seen us in the innovation room yet, please come, stop by, and let's have a conversation. Thank you very much.
Next, we have Aaron Lazarus with Encephial Therapeutics.
Hi, my name is Aaron Lazarus, and our team started Encephial Therapeutics. Our goal is to break the cycle of addiction using next-generation neurotherapeutics. There are currently no FDA-approved treatments available for cocaine and methamphetamine. The most successful and efficacious attempted therapeutic candidates were just too addictive to move forward. So there's a need to develop an effective treatment for cocaine and methamphetamine with little to no abuse liability. Our first-in-class treatment are new chemically modified analogs of the FDA-approved drug modafinil. They were patented by the NIH and exclusively licensed by Encephial. They were modified to increase affinity for the target transporter, the dopamine transporter, while at the same time keep a unique binding site that has proven to be non-addictive. We built our team by adding on an experienced addiction pharmacologist, as well as an advisory board with both regulatory and entrepreneurial experience. On top of that, because of the lack of the early expertise found in the pharmaceutical industry in this space, we partner with the experts found at the National Institute on Drug Abuse, a subset of the NIH, and Wake Forest, guiding us to bring this therapeutic to our exit point of proof of concept. With our work with our collaborators has already led to a successful tentative lead, as well as strong animal data showing efficacy and non-addictive tendencies. We have currently raised over $500,000 in non-dilutive funding, but we're hoping to complement that with seed investment to help finish our selection of our lead compound, as will provide us with strong drug development guidance. With this, we can achieve our goal of breaking the cycle of addiction. Thank you. If you have any questions, please come find us at our table in the innovation room.
Now we have Deborah Bitterfield of Lindy Biosciences.
Good morning, and thank you for coming back from the break for this. Lindy Biosciences is developing a formulation platform for biologics that will shift the standard of delivery from an IV infusion that takes hours to a single subcutaneous injection patients can administer themselves in less than 60 seconds, so a single shot under the skin. Now, biologics are an increasingly popular class of therapeutics, and for good reason. They're being developed to treat everything from cancer to autoimmune diseases to diabetes, and often with fewer side effects than other therapeutics. But they have to be injected. And a lot of these molecules, especially antibodies, require large doses, too large to fit in a single prefilled syringe. So they're given by lengthy infusions. But think about this. Every minute a patient spends with an IV in their arm is a minute they are reminded of their illness. It's a minute they are not out living their lives. For patients with chronic conditions, this can mean an afternoon every week just spent taking medication. We want to change that. Our technology uses a solvent extraction process to dehydrate solutions of biologics into a very dense powder that can then be injected as a suspension in a very small volume, and then it redissolves in the body. By using this approach, we more than quadruple the typical dose achievable in a single pre-filled syringe, and we have the added benefit of a more stable product. This is far more convenient for patients, it's cheaper for payers, and the pharmaceutical companies that develop these drugs gain a huge advantage in the market. We launched just over a year ago, and our dedicated team has been very busy. We are now generating revenue from service contracts and are testing with 2 of the top 5 global biologics companies, among others. We are currently raising our Series A round, and I invite you to help us get this technology closer to the clinic and closer to impacting patients' lives. Please come see us in the innovation room. We would love to speak with you. Thank you.
Please welcome Anthony Dellinger of Kepley Biosystems.
I'm going to take 2 minutes of your time today to talk to you about 2 innovations in interventional marine ecology. I'm your wild card today. What we've done is created organobait, a synthetic sustainable alternative to the use of wild fish for attracting crabs and lobsters to traps. Our customers are the commercial, recreational, crab and lobster fishing industries. Their problem is forage fish depletion is evident in all oceans. Thus, their costs are rising and their availability is diminishing. They have an urgent need to keep their sector profitable and operating. We've received over $1 million of investment from the National Science Foundation and the state of North Carolina. We've developed a product and it's ready to go to market. We don't need to use fish to catch fish anymore, and the industry needs this solution. With your help, we're ready to commercialize organobait, which mimics the molecules that forage fish release to attract crabs and lobsters to traps. It's sustainable, it's safe, it doesn't require storage, and it will not spoil. This is a $20 billion global bait industry. My second innovation, more relevant to this conference, involves the horseshoe crab. The horseshoe crab is comprised of a vital blood component that recognizes endotoxins. It's used as a raw material in a $120 million market space that's a $1 billion finished goods area. The problem is, the way we're bleeding horseshoe crabs right now is unsustainable. It results in the demise in 1 in 4 crabs. Our technology can humanely husband these species, safely bleed them, creating more LAL with 0 impact on the species. By creating a robust supply of LAL, we want to take this to the bedside, introduce a method for physicians to detect sepsis early using this molecule, arm the physicians with the information they need to start treating patients immediately. I'm table 13 in the innovation room. Come check us out. Thank you.
Finally, we have Tim Gallagher with PhosphoGamma.
I'm Richard Lopez. I'm not Tim Gallagher. My name is Richard Lopez. I'm a physician scientist at Duke University, where I'm a specialist in bone marrow transplantation and the cellular immunotherapy of cancer. In just the last several years, the field of cellular immunotherapy has become an extremely hot space, particularly within the sector of CAR T-cell therapies, as evidenced by a number of high-profile transactions, including a recent buyout of of Kite Pharma by Gilead for near $12 billion. However, even CAR T-cells, as promising as they are, still suffer from a number of major shortcomings, including the need for complex genetic engineering, the prohibitive cost of generating personalized patient-specific cell products for each and every patient, and most importantly, the extremely limited number of cancers that can be effectively targeted using CAR T-cell strategies. Indeed, despite the billions of dollars invested to date, CAR T-cell efficacy has only been demonstrated in selected patients with relatively uncommon blood cancers. Against far more common solid tumors, results have been disappointing at best. We believe the next generation of cellular immunotherapies must be simple to generate, inexpensive to manufacture, and above all, active against any and all cancers. With this huge niche in mind, I founded PhosphoGamma, a company focused on using human gamma delta T cells obtained from healthy donors to treat patients suffering from any type of cancer. Gamma delta T cells are unconventional T lymphocytes, which, unlike conventional T lymphocytes, can instantly recognize and kill any type of human cancer cell. In essence, gamma delta T cells are universal killer T cells. These cells are extremely rare. However, we've developed proprietary technologies that allow us to simply and economically manufacture practically limitless numbers of gamma delta T cells obtained from the blood of any healthy human donor. PhosphoGamma's approach is based upon creating a repository or bank of gamma delta T cells derived from a multitude of healthy donors. It is these banked universal killer cells that can then be used off the shelf at any time later to treat any individual diagnosed with any type of cancer. In summary, built around gamma delta T cells, we have developed the next generation cellular immunotherapy, which is simple and economical to manufacture, is truly off the shelf, and above all, is active against any and all human cancers. Given this, we are well positioned to challenge and eventually supplant the current generation cellular therapies, including CAR T-cell therapies, as we carve out a progressively larger and larger niche in the billion-dollar cellular immunotherapy market. We look forward to meeting potential partners and investors, as well as marketing experts and advisors who can help us advance this truly revolutionary platform technology. Thank you for your attention.